Hello, and welcome to Beyond Biotech, the weekly podcast from Labiotech. I'm Dylan Kassane, and this is episode 194 for the podcast. Today, we're joined by Patrick Andre, Chief Scientific Officer at Diagonal Therapeutics. A trained vascular biologist, Patrick's career spans groundbreaking work at Pfizer, Acceleron, Pliant Therapeutics, and earlier companies, where he focused on TGF-β superfamily signaling and receptor pathways that keep blood vessels healthy. Now at Diagonal, he's leading a bold mission, developing clustering antibodies that correct the root cause of serious genetic vasculopathies rather than just managing symptoms. In this episode, Patrick walks us through his personal journey into science, the company's Diagonal platform, and their lead program, DIAG-723, which recently received orphan drug designation for the rare disease HHT and is advancing towards the clinic. We also discussed Diagonal's oversubscribed $125 million Series B financing round that closed in January 2026 and what clustering antibodies could mean for patients with HHT, pulmonary arterial hypertension, and more. Patrick, welcome to Beyond Biotech.
Thanks, Dylan. Thanks for having me, and it's a true pleasure to be here.
To kick things off, maybe you could share a little bit about what first sparked your interest in science.
Really interesting question. I think there were probably two decisive moments. One actually that happened a long time ago, like 40 years ago, that was really painful, and one more recently that was really pleasant. So I'll go through the painful one, and that relates to broken ribs. When I grew up, I was, a kid really into comics books, science fiction, reading the Titan Marvel novels and all those things. And I was one of the kids thinking that if you concentrate enough, you can achieve fantastic things. So one day I was waking up, I tried to fly and I jumped out of the bed. And the landing was really pretty hard. And I broke two ribs. And that was the first time I really realized that things happen for a reason, and that science fiction actually was written with two words and not just one. So that was my first eye-opening moment that actually things are ruled by some laws, basically. The second moment came later when I was doing my postdoc at the Center for Blood Research. That was at HMS, and my PI was really fantastic, was Denisa Wagner. And this was the it moment for me for science. So it all came from an experiment I did in a dark room where we were doing intravitreal microscopy. And it consists in isolating cells that circulate in blood. And you wash them and you label them with a fluorescent dye. And you reinfuse the cells into a living animal. And when you use inverted intravitreal microscopy, it allows you to track interaction of circulating cells with the vessel wall. And it also allows you to, at that time, understand that cellular biology interaction of ligand protein with receptor that dictate activation of cells mediate actually a physical outcome, which is, for example, recruitment, rolling of platelet and leukocyte on the endothelium, adhesion or aggregation. And for me, it was fantastic because it was the first time I saw that cell biology was actually related to physiology in general and how organs works. And I was also exposed at that time, not just to the biological aspect of activating cells, but also the key importance of dynamic force, which are called shear force and shear stress in the blood vessel, which also dictate the response of the endothelium. So that was a late revelation, but that's what really drove me towards a scientific career.
So you did this training as a vascular biologist, but then you went on to work at some pretty important companies, Pliant, Acceleron, Pfizer as well. Walk me through your career so far.
Yeah, so yes, I was one of the few leftover scientists in France trained directly at being vascular biologist. I did my PhD at the Vessel and Blood Institute in Paris, which was dedicated to vessel wall and interaction of blood cell with the vessel wall. And so after that PhD, I moved to the Center for Blood Research. And there I had a unique chance to be exposed to many therapeutic areas because if you understand, blood vessels touch all the organs. So therefore, I was looking at blood-brain barrier, development of Alzheimer, for example. I look at atherosclerosis, thrombosis, coagulation, but also inflammation, rheumatoid arthritis, allergic asthma, and so on. So this was really fantastic because I applied my training in vascular biology to mechanisms of action that were going across multiple organs and disease. And the last part of my postdoc, we had this work collaboration with David Phillips, who was the founder of Pliant Therapeutics. And we work on the integration and interplay between thrombosis and inflammation. And so at the end of my postdoc, I joined this company. So I moved to the West Coast, and I spent basically 12 years studying vascular pathologies between Core, Millennium, and Portola. Same group of people, but 3 different names, which is typical of biotech. So that was my first experience in biotech. After 12 years, I moved back northeast. I joined Merck at that time, the cardiovascular disease department. And this was really interesting because I learned something totally different, which was the key aspect of translational pharmacokinetic, pharmacodynamic assessment, and the value of in vitro, in vivo correlation as part of key process for drug development. After 4 years, I moved back to biotech industry, first with Pliant and then with Acceleron. For both companies, I was looking at ways to modulate the biology that is associated with the TGF-β superfamily of ligand and receptor. And so I did that first at Pliant in the context of fibrosis, and then at Acceleron in the context of pulmonary arterial hypertension, and after that, HHT, which is hereditary hemorrhagic telangiectasia. And more recently, 2 years ago, I joined Diagonal Therapeutics, where we work on PAH and HHT.
Tell me about joining Diagonal. What made you say yes when you had the chance to come on?
Oh, it was first an easy decision, easy choice, because it's directly my alley, basically. It's vasculopathies, that's my training. And there was also something really interesting when I was a long time ago, that was back in 2019, started working on PAH and HHT. The 2 diseases didn't have a lot of drugs recently that were approved, and HHT still doesn't have any drug approved specifically for this disease. So it's a fantastic clinical unmet need. And on top of this, the therapeutic approach that we are proposing could constitute a pharmacological cure to the vasculopathy. And that made the difference, basically. To be able to make a difference in the patient life is fantastic.
Let's talk a little bit about Diagonal. For listeners who might not be very familiar with what the company does, give me an overview of that Diagonal mission.
So I think the main mission that we have is really to offer a pharmacological cure to vasculopathy and genetic disease in general. We are aiming at tackling the root cause of the disease. So we are not going after secondary pathways that are downstream, for example, the defect. We are going exactly where the defect is and bypassing the defect to provide a normal biology, a normal signaling pathway. This is what we are doing. And to better understand the mission, I probably need to spend a bit of time on describing what the disease is and why we're going after this disease with this specific modality. So for HHT, hereditary hemorrhagic telangiectasia, it's a vasculopathy that leads to a bleeding disorder, and that's driven by loss of function mutation in receptors that are present on the endothelium. So again, the endothelium is that layers of cells that face the blood and the vessel wall. And what happens when you have those loss of function mutation, you basically lose the vascular quiescence and integrity of the endothelium, and you form fragile capillaries that are prone to rupture. And that leads to hemorrhage. And so the manifestation that you can see from the patient is that they develop skin telangiectasia, but telangiectasia can also be present in the nose and the GI tract and lead to frequent and severe bleeding. Think about epistaxis, nearly 100% of the patient will have frequent nosebleed. And that leads to anemia in more than 60% of the patient develop a need for iron infusion and red blood cell transfusion. But the disease is more than the telangiectasia and the nosebleed. the patient also have a defective capillary system network, and they develop what is called arteriovenous malformation, which will form in different organs. So to give you a number, in the brain, about 20% of the patient will have brain AVM, arteriovenous malformation. About 70% of the patient will have AVM developing in the liver, and about 50% in the lung. And what you can clearly imagine is that if you suddenly have rupture of those blood vessels, then you're talking about severe morbidity and mortality. So the paradox is that it's a really severe disease. It's an orphan disease, but you still have a large number of patients. We estimate between the US and the EU that is slightly above 330,000 patients having this disease. And there is not a single drug approved for the disease. So this is really a conundrum because people know that there is a huge unmet need, but so far treatment that have been used off-label are typically treatment that will try to manage the bleeding and with repurposed drug, for example. So for us, the approach that we had was to better understand how to treat and make a significant difference is to go after the cause of the disease and to use for this clustering bispecific antibody, which will force association of receptor, bypass the deficient signaling to promote normal signaling, and basically really correcting the root cause of the disease.
Let's dive into a little bit of that science then. You mentioned this clustering there. Can you maybe explain in pretty straightforward terms what clustering antibodies are and how they differ from a more traditional antibody therapy?
Absolutely. So clustering antibodies basically do more than just a regular antibody that would be binding a target, whether a ligand or receptor. So they act differently than traditional antibodies. Their objective is to bring together a receptor on the surface of a cell in a very controlled way. And the ultimate outcome is to recapitulate what normally would happen with a normal complex of receptor that provide an important signaling pathway inside the cell. So at the endothelium, normally what you have is a ligand that will first bind a receptor, which acts as a co-receptor, it's endoglin, and endoglin at that time bring the ligand to a pair of two receptors which are ALK1 and two receptors which has BMPR2. And the evolution over the tens of thousands of years made this process really interesting and complicated because it's a key process for survival. And so once you have the ligand coming in contact with the tetramer, it will force association of the two ALK1 and the two BMPR2. And this association will allow the BMPR2, which is autoactivated, to activate ALK1, which is the signaling pathway. So the clustering antibodies are key because they will mimic the endogenous ligand and force the association of the receptor to promote the normal signaling. And only clustering antibody can do that because binding antibody will just bind one receptor, but will not carry the functional activation that has been designed by the nature.
What's interesting is you've got there at Diagonal a product engine that combines sort of the computational and the experimental techniques to discover these multi-specific antibodies. Can you walk me through how that platform actually works? And is it faster than doing it another way, or is it just more efficient? What's the benefit?
I would say it's both faster and more efficient. We know for a fact that in a 6 months time frame, we'll identify those antibodies, bispecific antibodies, and we'll have determined that they promote clustering of receptor and also that they carry the normal signaling biology in the relevant primary cell. And that is basically taking us 6 months. And the reason it's faster and more efficient is because we use computational technique that we combine with experimentation. And the computational technique is basically notably us working with an algorithm that was developed by Dr. Dima Kozakov from the University of Texas at Austin, which allow us to go through basically using AI technology about billions of combination of epitope that you will have on the extracellular domain of those four receptors, the two ALK1 and the two BMPR2, to also identify the epitope present on the antibody in order to have the best architectural association of those receptors that will drive activation of ALK1 and signaling pathway. So this is really fantastic because we are basically using this algorithm and computer to force association and provide us with likelihood of signaling pathway. And it allowed us to generate only a fraction of those combinations. So we typically end up generating 200 to 400 antibodies, which are representative of the key epitopes that are involved in the signaling. And we test them, and we usually have about 30% of those antibodies that are providing the desired signaling pathway. The next step after that, of course, is optimization of the antibodies, notably working on conformation, linkers, flexibility, and other point mutation that we add. So this is really a combination of AI and technology together with back and forth experimentation to confirm activity, and that goes really fast.
One of the key ideas seems to be this restoring of the receptor signaling, bringing the receptors together in a very specific configuration. Why is this approach useful when it comes to diseases like HHT, like pulmonary arterial hypertension? Why is it so effective?
So it probably comes from the real importance of the biology of the vessel wall across all the organs during development and adulthood. This is what allows you to be alive, basically. And so what it requires is a precise machinery that will maintain integrity of the endothelium. And the precise machinery sometimes will not utilize just a single receptor and an interaction with one ligand, but a combination of receptor and ligand in this precise succession. And so what you have is in HHT1, which is a loss of function mutation in endoglin. Endoglin is the co-receptor, which is the first one to bind the ligand that normally activates the endothelium and provide vascular quiescence. So if you only have one receptor, then you have less binding of the ligand to the receptor and less signaling. And so you also have in HHT2 a loss of function mutation in ALK1, and here again, only one of the two receptors is active and you lose signaling. That's what leads to the loss of vascular integrity and the development of those AVM. So this interplay is depending on the precise location of the ligand on key epitopes that are present on the extracellular domain of 2 ALK1 and 2 BMPR2. And then it also requires in the cytoplasmic domain of the receptor, which are present inside the endothelial cells, to have a close enough proximity so that BMPR2 can activate ALK1. And this happens only if you cluster the type 2 receptor with the type 1 receptor in a specific manner. Otherwise, you don't have activation. And this is the reason why you have a disease associated with only 50% downregulation. And that disease leads to fragile blood vessel and hemorrhage.
One of the things that comes to mind when I hear you talking about this approach is it seems like it all makes sense. It all falls into line. The optimization, the AI, the computation, the experiments, and then we have this fantastic solution. But I assume that there's got to have been some pretty technical challenges along the way that you've had to overcome. Tell me a little bit about that.
Yeah, and it's part of we are trying to solve a problem related to, let's say, loss of function mutation, which are disrupting the normal biology. And that normal biology has been established over the course of tens of thousands of years. And that has been, I would say, the method and biological pathway that Mother Nature created inside the body to make it work. So the complexity that we face is that when you bring in, for example, a loss of function mutation, a pathogenic mutation, it creates an evolutionary unprecedented search space. So, and that basically affects the ability of normal generative AI design methods to identify the corrective antibodies. So the challenge is you're working now with a biology that is different than the logical one that has been put in place. And so this is the biggest challenge because you're going into the unknown. And so the beauty of using this algorithm is to be able to systematically explore all just that position of antibody arms and receptor complex computationally, because you would not be able to do that if you were to have to make 100,000 different antibodies. It would be too complicated. So we use basically a software to appreciate the likelihood that those pairing of location on the receptor and the antibody will drive the biology. And that's feasible only because of this.
In preparing to talk to you today, I did some reading and I saw in the preclinical data, it indicated your approach can prevent and even reverse the disease pathology in certain models. Have I got that right? And, if that's right, can you tell me a little bit about how that would look in practice?
You're absolutely right. What we have done is we use several translational models of HHT in collaboration with Philippe Marambo from the Feinstein Institute. And we have demonstrated with DIAC-723, our lead program, that we can indeed prevent and reverse establishment of AVM, both in mice and rat. We also have demonstrated preclinically that we can reverse anemia, and we can prevent mortality due to excessive blood loss. And we also perform experiment using human cells from patients who have those loss of function mutation and confirm that our biologics clustering antibody promote clustering of the receptor and normal signaling pathway. So what does that mean for the patient? So we believe that DIAC-723 has actually the potential to become a disease-modifying treatment, and that it will, over time, provide vascular quiescence in patients. So it will translate into a progressive reduction in frequency and severity of epistaxis. We expect to have correction of anemia, and also, over time, resolution of vascular malformation, as the blood vessel possesses a strong intrinsic plasticity to remodel. And we will notably, during the clinical development program, we'll have an open label extension study where we'll be looking over time at resolution of those established AVM, which will allow us to document that we can indeed reverse in clinic. And this will happen pretty soon because we are filing with regulatory agencies right now. We are about to enter the clinic in mid-2026, and we are looking forward to assessing the potential of our drug.
Let me pull you off the science for a second and into the business side of Diagonal. You closed an oversubscribed $125 million Series B financing in January. What does that mean for the company right now to have that sort of confidence being thrown your way?
So there's two aspects there, I would say. So the financing was led by Sanofi and Janus Anderson. And what's really great is that, of course, it brings in dollars, which allow us to accomplish our objectives. But it also adds scientific expertise to the group. We found partners, actually, who can also help us think about development of our drug, the disease, and they have a really strong experience in cardiovascular disease as well. The proceeds from the financing will enable us to advance our lead program and to deliver multiple clinical proof of concept readout for DIAC-723. The first one will be in HHT, but we will also look at a subpopulation of HHT patients who also develop PAH. And we also have another indication where we think that DIAC-723 can make a difference. And also what's really important is that it will allow us doing the SAD, which will be initiated directly, so SAD for single ascending dose, which will be initiated directly into HHT patient. We will establish, of course, safety, but we also have identified specific biomarker that track with the disease and also that are sensitive to the mechanism of action of our drug. So we expect to generate proof of concept data by the end of the year, at the latest, the beginning of 2027. But the money will also allow us to move forward. We have 4 other programs in early discovery. Our objective is to achieve drug candidate nomination by the end of 2026 and to move at least one new program into IND filing by the end of 2027.
Talking of regulation, you've got orphan drug designation for DIAC-723 from the FDA and the EMA for HHT. How important is that sort of support as you're moving forward into clinical trials?
Well, I think it's a reflection of the unmet need. The process went really smoothly, and since I emphasize it's a severe disease associated with morbidity and mortality, and there is not a single drug that has been approved, no phase 3 trial actually, for this specific indication. So it also reinforced the potential of DIAC-723 to really significantly improve the lives of people and families that are affected by the disease. So it's a really, to some extent, it's not surprising that you would have ODD from both the US and the EU, because there is a fantastic unmet need and there is a chance to make a difference.
There are other companies that are working on these rare genetic diseases and even with antibody-based therapies. How does your clustering antibody approach stand out from what they're doing?
First, I have to say that it is great that there are more than one company working on HHT, because the unmet need also, we stimulate each other, we look at our different mechanism of action, and we think, for example, that we have an approach that will be the one that can potentially provide a cure pharmacologically to the disease, because we target the root cause of the disease. But there is always room for other mechanism of action that eventually would be complementary. But the beauty of our approach is that we really go at the source and we target where you have the defect. And based on the experiment that we generated, we see that we correct the entire defective signaling pathway. Most of the other drugs that are either being used off-label are typically managing symptoms. Some of them are repurposed drugs that were developed for angiogenesis/oncology. Others are being under development for HHT, but they target more downstream signaling pathway. And based on this, I'm not suggestive of full correction of the defect.
Let's look forward now. If you're looking ahead, how do you think advances in this clustering antibodies and the receptor signaling therapies could reshape the treatment landscape for genetic disease, maybe if we're looking forward 5-10 years?
So if we think, for example, about HHT, it's really a genetic disease driven by a loss of function mutation, 2 different receptors. But our approach could be a safe equivalent to gene therapy, I would say, because those mutations on either ligand or receptor can be targeted by well-validated modality, which is basically biologics. And there are more than 50 years now of experience with development of biologics. And it's relatively easy to monitor and understand how it works. The safety profile usually is associated with on-target activity. So I would say that if you think about the novel technology around gene writing, gene editing, I'm not there yet. It's interesting, but in the context of mutation of ligand and receptor, notably if clustering of receptors are needed to promote a key signaling biology process, our approach is probably the most logical and safest and easier to actually tackle, because we really go after what is being dysregulated.
What are some of the trends that you're keeping an eye on as we're moving forward into this new era of therapies?
That's it's a really interesting question. We look at about everything, of course, related to competition, different modalities, different target, but also the waves of science that are coming from, both the US and other countries. We pay special attention to China's rise in biotech innovation, for example, application of AI in drug development, but also drug discovery. We do that internally as well. There is also fantastic press around novel design of antibodies. Recently, we talk about ADC, but you have bispecific, trispecific antibodies. And now people are also trying to increase, for example, biological half-life of those antibodies, as well as other modalities. So we pay attention to everything because it is required for us to know what's coming and how to position ourselves. But I would say everybody does it. We have to pay attention to and learn from what others are doing, because that's what allows science to move forward.
Finally, Patrick, for the listeners who want to follow along with your progress at Diagonal, learn a little bit more about the science and the company, where's the best place for them to go?
I would guide them toward our website. It's diagonaltx.com. You can also find information on LinkedIn at Diagonal-Therapeutics.
Patrick, thanks for joining us today on Beyond Biotech.
Thanks for having me, Dylan. Have a good day.
That's it for this episode. Don't forget to check out all the latest news and analysis at Labiotech.eu. I hope wherever you are in the world, you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Beyond Biotech.